Coming to the next phase is S phase. S phase is also called synthesis phase. S phase is also called synthesis phase, right? So in this phase, DNA synthesis takes place. This one is also called synthetic phase. This one is also called synthetic phase. S phase is also called synthesis phase or synthetic phase. In this phase, the DNA synthesis takes place, right? See, within S phase, DNA replication takes place. DNA replication takes place. DNA replication is nothing but this is the one of the cell. This is the nucleus. Within the nucleus, DNA content is present. DNA content is present. That DNA content, okay, that DNA content in the normal body cells, the state is the 2n state. The state is the 2n state. After the S phase, or end of the S phase, the 2n becomes 4n. End of the S phase, the 2n becomes 4n. So here what I am telling, there is a G1 phase is there, S phase is there, G2 phase is there, right? See, within G1 phase, the genetic material content is 2n. Okay, in the S phase, early S phase, the content is 2n, but end of the S phase, the content is 4n. The DNA content is the 4n. And coming to the G2 phase, the DNA content is the 4n. The DNA content is the 4n. So there is an S phase is there. Within that S phase, there is a DNA replication takes place. Within that DNA replication process, whatever the genetic material is there, the genetic material will be doubled. The genetic material will be doubled. Remember, not the starting point of the S phase. Okay, end of the S phase, the genetic material will be doubled. End of the S phase, the genetic material will be doubled, right? So that is the one of the important point we discussed. Within G1 phase, whatever that proteins are there, which are nothing but enzymes, the enzymes involved in DNA replication process, right? So that is about the DNA replication. This is the DNA content. Later we will discuss about prophase, metaphase, anaphase, how the chromosome number is varied, right? So if this is about the G1 phase, the genetic material content is 2n. S phase, the genetic material content is 4n. In G2 phase, the genetic material content is 4n, because here the genetic material is doubled, right? Coming to the next point is the histone protein synthesis. Histone protein synthesis. What are histone proteins? Then what is the function, or what are the functions are performed by the histone proteins? What are the functions are performed by the histone proteins? Within the cell, okay, the proteins, means histone proteins here synthesized in cytoplasm. There is an H1, H2A, right, H2B, H3, right, H4. These are the types of histone proteins. These histone proteins are involved in the condensation of genetic material. That is nothing but this is the DNA content. That DNA content majorly forms the chromosome structure. Presence of histone proteins. So presence of histone proteins, the DNA content, this state is called as chromatin. Chromatin is converted into chromosome. Right? So chromosome are chromosomes, right? So here chromatin is converted into the chromosomes carried out by the histone proteins. Remember, not only histone proteins, some of the non-histone proteins also present. But for the chromosome formation, major crucial role done by the histone proteins. Histone proteins are there. Histone proteins are majorly involved in the chromosome formation. We already discussed how the histone proteins have the capacity to interact with the DNA. how the histone proteins have the capacity to interact with the DNA. Histone proteins are having positive charge. Histone proteins are having positive charge. Histone proteins are having positive charge. DNA having negative charge. DNA having negative charge. Here, why histone proteins are having positive charge? Within that histone proteins, lysine amino acid, arginine amino acid, lysine and arginine amino acids are present. Presence of lysine and the arginine amino acids, right? The histone proteins are having positive charge, and the DNA, here DNA presence of phosphate, So presence of phosphate, the DNA having negative charge. So opposite charges are present. Because of these opposite charges, both these are combined and forms the chromosome structure. For the chromosome formation, non-histone proteins also supported. Right? So here question is generally DNA. Here question is generally DNA present in nucleus. This is the DNA content. Right? See here cytoplasm. This is the cytoplasm. Protein synthesis takes place. Chromosome formation takes place inside the nucleus. Then, sir, histone proteins are synthesized in nucleus or cytoplasm? First question. Histone proteins are synthesized in nucleus or cytoplasm? No doubt, always protein synthesis takes place in cytoplasm. Right? So, histone proteins are synthesized in cytoplasm. Which are transported to. The histone proteins are transported to inside the nucleus. So this is a very easy concept. The histone proteins are synthesized within that cytoplasm after the histone protein synthesis process, which are transported from cytoplasm to nucleus. Then these histone proteins are interact with the chromatin, finally forms the chromosomes. So within histone proteins there is an H1, H2A, H2B, H3 and H4. These are the types of histone proteins are present. These histone proteins are majorly involved in the chromosome formation. We already discussed this kind of chromosomes are called as sister chromosomes. Right? Right? So that is about the histone proteins and histone proteins function. Right? Coming to the next one is centrioles replication. See, centrioles, this is one of the cell contains within G1 phase, there is a single centriole is present. This is the single centriole. Once the cell enter into the S phase, the centrioles are replicated. One centriole forms the another centriole. So here centrioles formation means already one centriole is there. Right? The second centriole formed during S phase. So centrioles replication. Simple. It's like a DNA. DNA. This is the 2-end state. Right? In S phase, the DNA becomes 4-end state. So that is nothing but DNA replication. Already in one cell, one centriole is present. That centriole involved in replication process means another centriole is formed. So the step is called as centrioles replication. So once the centrioles are replicated, this structure is called as centrosome. We know this structure is called as centrosome. Centrosome contains 2 centrioles. So during cell division process, these centrioles, always these are present perpendicular, right? These centrioles are moving towards the opposite pole side. And which are involved in the spindle apparatus formation. Which are involved in the spindle apparatus formation, right? So this is about the spindle apparatus. This is about the spindle apparatus. Once the spindle apparatus are formed, which are connected to the chromosomes, then chromosomes are pulling towards the opposite pole side by the spindle apparatus. So here centrioles replication takes place in S phase. So in all cells, centrioles replication takes place in S phase. In all cells, listen question carefully. In all cells, centrioles replication takes place in S phase. Definitely I will say my answer is no. Because all cells are not having centrioles. All cells are not having centrioles. This point, okay, related to only animal cells. The centrioles replication takes place in only animal cells. Right? So here, whatever the animal cells are there, the animal cells contain centrioles replication only takes place in S phase. Remaining all cells are not having that centrioles. So centriole replication is only seen in S phase. Centriole replication is only seen in S phase and animal cells. Only seen in animal cells in S phase. Right? Only seen in animal cells in S phase. Right? That is about the S phase. That is about the S phase. Coming to the last one, G2 phase. We'll discuss about the G2 phase. Right? Coming to the next phase is G2 phase. In the interphase, the last phase is the G2 phase. See, the very first phase is called as gap 1 phase. Because of the DNA synthesis, the next phase is called as S phase. The last phase is called as gap phase within interphase. Right? So that is about the G2 phase. Here G2 phase is also called post DNA synthesis phase. Post DNA synthesis phase, because after the DNA synthesis, the G2 phase is present. That's the reason why this one is called as post DNA synthesis phase. Right? And this one is also called pre-mitotic phase. Pre-mitotic phase. Simple. Okay, after the G2 phase, the next phase is mitotic phase. After the G2 phase, the next phase is the mitotic phase. So this phase is also called pre-mitotic phase. This phase is also called pre-mitotic phase. Right? So that is about the introduction for the G2 phase. Right? See, within G2 phase, protein synthesis is increased. More amount of proteins are synthesized. I already said the very common point is in all these phases, protein synthesis takes place. Right? So more amount of proteins are synthesized. And next one is tubulin protein synthesis takes place. Sir, initial way the tubulin proteins are present. I thought how many number of students are known about the tubulin proteins. See, this is about centrioles, right, which are involved in the spindle apparatus formation. So the spindle apparatus formation majorly done by the tubulin proteins. So these are nothing but tubulin proteins. These tubulin proteins are polymerized. Then which forms the long thread-like structures. Okay, these are called as spindle apparatus. Remember, within the cytoplasm, the tubulin proteins are produced. Within the G2 phase, these tubulin proteins are polymerized by the help of centrioles. Right? See, once the polymerization takes place, these are forms the long tube-like structures or long thread-like structures, not tube, long thread-like structures, right, which are involved in the pulling of chromosomes towards the opposite pole side. So the tubulin proteins, here more amount of proteins are synthesized. The proteins are nothing but tubulin proteins. In S phase, more amount of proteins are synthesized. The proteins are related to histone proteins. In G1 phase, more amount of proteins are synthesized. The proteins are related to DNA polymerases Okay, that is nothing but involved in the DNA synthesis. Involved in the DNA synthesis. Not only DNA polymerases, RNA polymerase also synthesized. Okay, and topoisomerase also synthesized. So the enzymes which are involved in the DNA replication, all these enzymes are synthesized. Here enzymes are nothing but proteins, right? RNA synthesis is increased. Here RNA synthesis is increased. We know RNA synthesis increased, protein synthesis also increased. RNA synthesis increased, protein synthesis also increased. So here more amount of RNA synthesis takes place. And next one is ATP synthesis. In a cell always ATP synthesis is very important. ATP synthesis is very important. So here in a cell more amount of ATP synthesis takes place, then all metabolic activities takes place. Remember, for the cell division, the very important factor is the energy. Very important factor is the energy. Okay, if energy is available, the cell division takes place. If energy is not available, the cell division will be halted. Halting is nothing but stop. So within that G2 phase, protein synthesis, tubulin protein synthesis, especially, right, RNA synthesis, more amount of RNA synthesis takes place, and more amount of ATP synthesis takes place. And one of the important point, but the point is contradict. In some books are given in one way. In some books are given another way. I like to discuss this point, but I'll give that clear statement. See, cell organelles are doubled. So here cell organelles are doubled. So here my clear statement is cell organelles are doubled in which phase? Some books given in G2 phase. Cell organelles are doubled, so number of mitochondria are doubled, number of within plant cell number of chloroplasts are doubled. Okay, centrioles here doubled, right? So what I'm telling here, whatever that the cell organelles are there, whatever the mitochondria, endoplasmic reticulum, Golgi complex, right? The cell organelles are doubled in G2 phase. In some books is given, but according to the NCRT, we have to follow the cell organelles are doubled in G1 phase. Both the some books, maybe in the future some students searched in internet. If the student asks me, sir, you said like that, but here given like this, that time I have to say the answer. Right now if I explain, I don't face that problem. That's the reason I am saying some books are given like this, The number of cell organelles are doubled in which phase? No doubt G1 phase. The phase is the G1 phase. See, some books are given like this, but we are not following this one, we are following this one only. So number of cell organelles are doubled in G1 phase. Sir, you said in interphase the cell size is increased. So in which phase the cell size is increased? I said in interphase the cell size is increased. In which phase the cell size is increased? Here also answer is very simple. We are following this one. When cell organelles are doubled, the cytoplasm content is increased. So the cell size is increased here. In which phase? G1 phase. Then add these 2 points to the G1 phase. So cell organelles are doubled and cell size increased in G1 phase. Right? So that is about the interphase. Remember, interphase is also called preparatory phase. Why this one is called as preparatory phase? Remember, here enzymes are synthesized for DNA replication. So DNA replication takes place. Here centrioles replication takes place. Histone protein synthesis takes place. So histone proteins are supporting for the chromosome formation. Centrioles are supporting for the spindle apparatus formation. So here tubulin proteins are formed. which are involved in the spindle apparatus formation directly, right? So here ATP synthesis. So during cell division process, energy is required. So whatever that components which are required for the cell division, all these components are synthesized in interphase, right? Interphase prepares all the components required for the cell division. In mitotic phase, there is a karyokinesis process, cytokinesis process will be takes place. Karyokinesis and cytokinesis process will be takes place. That is about the interphase. Next one we will discuss about the mitotic phase, right?